Oh. All right, let's go back and watch the whole thing again.
To sequence DNA by the chain termination method, the DNA must first be obtained in single-stranded form. A mixture containing the single-stranded DNA, DNA polymerase, the 4 deoxyribonucleotides A, T, G, and C, and a single short primer is prepared. The primer has a nucleotide sequence that is complementary to the 3' end of the region to be copied, and is required so that DNA polymerase can initiate DNA replication. Equal amounts of this reaction mixture are placed in each of 4 tubes, and a different dideoxyribonucleotide is added to each tube. When a deoxyribonucleotide is inserted into the growing chain, replication continues. However, when a dideoxyribonucleotide is inserted, strand synthesis is terminated. The contents of the reaction tubes are then transferred to 4 lanes of an electrophoresis gel, and the oligonucleotides are separated by size and nucleotide type. The shortest oligonucleotide moves furthest down the gel. Reading from bottom to top, 1 base at a time, provides the correct DNA sequence.
Does that make sense? You see how the one at the bottom is the shortest, and then there's a G. And the reason why each one stopped at that point is where that DNA polymerase randomly put in a dideoxynucleotide. So the lab we're going to do today is on finger sequencing, and you get to like do this with copies.
Also like what the primer is supposed to be.
What?
How do they know what the primer is supposed to be?
Well, they would have to know a little bit about the sequence.
Okay.
Yeah. Otherwise, it wouldn't work. Okay. So real fast before I move on. So the old gels would look something like the picture on the right. When you use radioactive dideoxynucleotides, you have to take the X-ray picture, and it looks something like that. And good God, imagine reading that. That would be horrible. Computers, they started to use computers, but then they also started using fluorescent tagging. So instead of using radioactive dideoxynucleotides, now what they use, there's the guy, yay, what they use are fluorescent nucleotides. And so you have no more radioactivity. You don't have to use the X-rays anymore. You also can load them all in the same lane because each letter will now fluoresce a different color. So you can all run it on the same lane and then read it all colored. Like, does that make sense, from the bottom up in one lane? So you can sequence a lot more samples at one time because you can fill a bunch of lanes. And each lane will be a different sequence. So it will look something like that. And computers will read that now, kind of like the microwave. You shine a laser over it, the computer gets the information. It has an output that looks something like that. Well, you wouldn't have to read it. The computer will tell you. It looks like that. And the computer would just tell you what the sequence is. It makes it a lot faster and a lot cheaper. And there we go. So why do we care about sequencing DNA? We'll do the lab in a minute. Because it gives us a lot of information to tell what your gene sequences are. A lot of human disease is caused by genetic mutations. And to know what actual genes should be, their sequences, and compare them to disease alleles and give you information about maybe what's going on. You can do research about what genes code for what proteins or whatever. The human genome was sequenced fully in 2003. They started the sequencing of the human genome in 1990. So it took around 13 years. They thought it was going to take 15 years. And they talk about this in chapter 21. I didn't make you read that part because I was going to talk about it. But computers are used to do a lot of the sequencing nowadays. And you would have these giant labs. And it takes a lot of, I'll show you a video where you can kind of see this stuff. Anyways, the picture on the bottom left is what like a Sanger sequencing output would look like in a computer. See all the lanes and the colors, and computers do all that stuff now.
It's crazy, like the people that are behind the technology stuff wouldn't get recognized by the mobile side or something without those You know?
Right. Well, they do still because the people. Yeah.
It's like
Yeah.
you only see like one person.
Well.
Coming up with something.
Yeah, it's a lot of. I bet the person who come up with the CRISPR might get something.
That's my main man.
President Clinton. So the Human Genome Project started as a governmental thing across the world. It began where scientists across the world were trying to sequence the human genome. And they were using a method to do that using this map-based method. So they would cut a certain piece of DNA into a bunch of different fragments and arrange the fragments based on overlap. And then they would figure out, okay, so this one overlaps with this one, so that means they must be right next to each other. But since like C overlaps with D but doesn't overlap with B, it must not be next to B. Does that make sense? Mm-hmm. This took a very long time because they only sequenced it by chunk by chunk by chunk at a time. And it took a relatively long period of time to do that. Along comes a guy named Craig Venter, who basically was like, hey, I can sequence the whole human genome in like 9 months. I don't need 15 years to do this. And he owned a company, or he was part of a company called Celera Genomics. And he is on the left in both of these pictures. Craig Venter, that was a young Craig Venter. But he worked for not a government. And his method of sequencing the genome was the shotgun method, where he took the whole chromosome, and not just a piece of it, but the whole chromosome and cut it off all at one time. And then cloned them and looked at overlaps that way. But instead of using just piece by piece by piece at a time and trying to figure out the sequence of just that piece, he did the whole chromosome at one time. And it did go much, much faster. So with his help, the government did sort of a joint conference, which is the picture on the top right, where they sort of tried to work together to say, hey, this has been sequenced. There's still a lot of bad blood. There's politics in science all the time anyways, but you'll see about that kind of stuff in some videos I'll show you. Yeah.
You know, I said, that it, like, is that the guy? Is that the W.S. guy?
Yeah. Yeah. So 2001 was they published this draft. 2003 is when it was finalized. And what it is that they sequence the genes. There's a lot of areas in the repetitive DNA and the junk DNA that haven't been fully sequenced because it's hard to do. If there's so many repeats, it's hard to figure out where one thing overlaps with another because of all the repeats. So those areas are still sort of trying to be figured out. So let's do our lab before we move on and talk about this part. I don't want to sit the whole time. We didn't do this in first block. We will. Okay. Friday, y'all need to get up and move. Really is. I feel like a lot of people aren't here today. Really? Yeah. My other class tomorrow was super easy. All right, so this lab, there's a bunch of information about Sanger sequencing. And then they talk about Sanger sequencing, so you can look at all that. Let me kind of skip to the part where you get to simulate it. So we're not exactly doing what it says because, one, you're going to be in more than 4 groups. If you turn the page, well, it says simulation, like turn for turn. It's on the back of the 3rd page, maybe? Yeah, over here. The back of the 3rd page. All right, so basically it says, down here it says teacher guide. Divide the class into 4 groups. I'm not doing that. You're going to be in a group of 2. Okay, you pick a partner, okay? So we're going to be way more than 4 groups. I will tell you and your partner which dideoxynucleotide you will have. You're not going to do all of them. Does that make sense? So I'll be like, you're going to have dideoxy A. So, you know what I'm saying? So y'all just, what you're going to do is if you turn it over, here's a little template sequence you're going to try to sequence. It's right here. Okay. So your template is right there. So if you're doing dideoxy A in your little reaction chamber, you're going to have a bunch of normal nucleotides, all 4 normal. So then you will have some, and it tells you the quantities on here, how many dideoxys you are. And then you'll read the directions, but you're going to synthesize the fragments basically where the dideoxy A's would all be put in. So if you have dideoxy A, how many fragments will you make? Look at it. 1, 2, 3. You have 3 different fragments. There's 3 different A's. One will be 1, 2, 3, 4 nucleotides long. One will be 10 nucleotides long, and one will be 12. See that?
Huh? Okay.
That's where the A's are. You have to count how many nucleotides.
Oh,
1, 2, 3,
and there's 4. So that's 12.
1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Does that make sense? Okay. Once you do all your little fragments, and how we're going to figure these hot beads, I'll explain in a minute. Then there's a giant gel electrophoresis chamber back there. The green little stickers are the ladders. You see that? So each nucleotide, and I wrote on there how many bases long. Like if you had 1 base, it'll go to the very bottom. And I wrote, when you go back to your see, but on the gel also, there's wells at the top. On the wells are yellow. I wrote A, T, C, and G. So if you have the dideoxy A, you're going to put your little fragments in the A chamber. You're going to put your fragments based on what size they are, where they would run out. Does that make sense? When you and your partners finish, you'll put your fragments in the gel. Got it? So then we're going to use the whole class. Once everybody's got there, then you're going to draw what the gel looks like on the gel here, and then you'll answer the questions. It's really not hard to do. And then you'll write the sequence out. So it's really not hard, but it's just to sort of get your hands and figuring out, or like get your brain and figure out what this is. It really will, if it doesn't make sense to you, it should make sense. Now, how do you set up your stuff? I'm going to give you copies. And it said to like put a piece of tape or something. It tells you on here with the top beads. The 5' end will be where the hole is, and the 3' end will be the little thing sticking out. So the hydroxyl group will be the thing sticking out. If you have a dideoxy one, put a piece of tape or something over it to indicate like the chain can't keep going. Does that make sense? All right, so follow the directions, how it says to do it. Okay, I didn't really explain all that. But just go ahead and run it on your little gel. Yeah, that'll be fine. There's gonna have to be a group of 3. Fine. Okay. We can have 2 groups of 3 or 3 groups of 3 if you want to do that. That would work. Okay. When you say your group, just come to me and I'll tell you what letter I want you to do. I'm going to go get the top piece out. Ready? I don't want this to take till the end of class. I want this to take until like maybe 12 maybe. Okay?
Wait.
What?
Same thing. You need to do this and do this. Here, you got a graphic right now. Say Cassandra. As long as they identify people, they figure
Copy. Y'all have your group?
Yes.
Here you go. Sign it. Okay, this one. D, A, B, C. Those are the groups. D, A, here you are. C, you with them? Go over there. C, put yourselves up. Go find 3 groups. Y'all group 3?
well, it's this.
Who are you with, Andrew?
I don't know yet.
No, all 3 of you go find a group. We're going to just have 2 of each letter. There you go. Here's how Yeah.